Hello viewers, welcome to Biodrunks. PDF link in the description.
In the previous video, we learned about vectors and we learned about what are the types of vectors. So from today's video, we are going to do one by one all the vectors and learn more about them in a very detailed manner. So the first vector which we are going to do today is plasmid vector. So before going into deep about the plasmid vectors, let's learn what is a plasmid. So plasmid is a DNA molecule which is present in the host cell other than the host chromosome. In a bacterial cell other than the bacterial chromosome. So for example, this is a bacterial cell with its chromosome in which you have your plasmid which is circular in shape. And remember, this plasmid is capable of independent replication, transmission, and its size ranges from 1 kb to 250 kb. Okay, so this was a brief description about plasmid. Now, what does this plasmid have? So, whenever you talk about a plasmid, okay, these plasmids are classified into several incompatibility groups. Okay, two or more different plasmids that belong to the same incompatibility group cannot reside in the same cell. Okay, so we have to first keep it in mind that the plasmids that belong to a single incompatibility group, they can share the same mechanism of replication control. So that means whenever you are using a plasmid, you need to mostly focus on the replication control. Okay, and always remember, these plasmids are not essential to the host cell except for certain environmental conditions. Okay, so that is the reason why we are using them as a vector. Now, there are different types of plasmids. So we can briefly differentiate them as F plasmid, R plasmid, and Col plasmid. So F plasmid, they are responsible for conjugation. So conjugation means F means fertility. Okay, this F word is derived from fertility. Okay, and it produces TRA gene which is used for conjugation. Okay, now R plasmid. So what is this R plasmid? They carry the genes for resistance to antibiotics. Okay, now for example, R plasmids, they are very important in human health. Okay, like they can make bacterial cell resistant to specific antibiotics. Okay, now some of them are provided that the antibiotic resistant markers are also there, and this is very important for gene cloning experiment. So one example of R plasmid is R4, RP4. I can write it here, RP4. So this is a Pseudomonas species. Okay, now you have col plasmid. These plasmids, what does they do is they code for colics. 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 The proteins, these are the proteins that kill sensitive E. coli cells. Okay, and they also carry genes that provide immunity to a particular clonics. Okay, so this was how we differentiated plasmids. Now you can also differentiate them on the basis of one of the most important point, that is conjugation. So when you talk about conjugation, they are differentiated as conjugative or transmissible and non-conjugative. So conjugative and non-conjugative. Conjugative means what is happening here? They mediate the DNA transfer, okay, through conjugation. Non-conjugative means they do not mediate the DNA transfer through conjugation. Very easy. Now, this was for plasmids. Now we have replication control system. So what is this replication control system? See, each plasmid is maintained in the bacterial cell at a characteristic copy number. It's due to the replication control system. So when you look at that, you have two types, single copy plasmids and multi-copy plasmids. So what is a single copy plasmids? It is same in the copy plasmids. The replication is same as that of the host cell, bacterial host cell. So they replicate and segregate with the bacterial chromosome. This is also called as stringent replication control. Now you have the next one as multi-copy plasmids. So what happens in multi-plasmid plasmid is these plasmids replication system is different from that of the host cells. So they undergo more than one replication. Okay, for each replication, for whenever the host cell is replicating, they have more than one replication. And hence it is called as relaxed replication control. Okay, so we know the two types of replication: stringent replication control and relaxed replication control, or you can also call it as single copy and multi-copy. Okay, now see, whenever you are using these plasmids as a vector, they are natural plasmids, so we need to modify them. We need to shorten them, reconstruct them, and we need to recombine them in vivo or in vitro. Okay, so that they can be used for a particular function. Now, whenever you talk about plasmid, E. coli is the plasmid which is being used mostly for the gene cloning experiments. Okay, now the earliest plasmid vector that contains the replication module ORI and a resistance gene, okay, that was PSC101. So we are not talking about PSC101 now, okay, because it does not permit direct selection of cells, okay, and it also contains unnecessary DNA and has stringent regulation of replication. Okay, so because of such factors, we are going to learn more about the mostly used vector, that is PBR322. So before that, what is a plasmid vector? It's a cloning small DNA fragment, okay, with small size, high copy number, origin of replication, restriction site, selectable markers, and so on. So as I said, we need to modify the vector because under its natural conditions, it won't help us. Okay, so what do? We insert multiple cloning sites. That means we need to have restriction sites. And these restriction sites should be common for a lot of restriction endonucleases. Why? Because when you're placing this particular DNA insert, okay, so this DNA insert has a particular restriction site which has been cut by a particular restriction and endonuclease. So it is cut by a particular restriction endonuclease. Now again, it should be fitting with the vector having cut by the same restriction endonuclease. Then only it would be fitting here, right? So for that, you should be having a proper knowledge about which is the restriction site. Now, antibiotic resistance gene. See, few host cells, they are sensitive to antibiotics. So whenever you're making a particular vector, you should be very clear with the resistance gene that you are placing with it. And also, when you are placing the resistance gene, they also act as a selectable markers. We will get to know it later. Now, not only that, you have to insert screening, selection systems for screening. Screening is very important. Okay, if you're placing a particular DNA insert in a particular gene, it makes it non-functional. Okay, why is it non-functional? Because it gets inactivity. That means if this is a sequence for a particular gene, for example, A, T, T, G, C, G, and you are just cutting the sequence, that means it is not working now. This doesn't code for a particular gene. Okay, so that's how you make it non-functional. Okay, now let's see how does your PBR322 looks. So as I said, it has a polylinker, antibiotic resistance, and selectable markers. And the most important is origin of replication. This is the most important of all. Okay. Now, see, we were just discussing that the markers are very important. And why the restriction enzymes are also very important. See, when you are cutting this particular, I should say plasmid, okay, so here the insertion site of the particular plasmid is within BLA or I should say TETZ, okay, and then you have cells that has a non-place site where you have ampicillin gene. Okay, now if I cut If I use a certain restriction endonuclease, and if I cut it and I insert the DNA, if this DNA is inserted in the ampicillin resistant gene, so what will happen? That particular cell will not grow in ampicillin. Okay, that means the vector has been transformed. If it is growing, the vector is not transformed. Similar thing goes for tetracycline also. Okay, this is PU18. P means plasmid, U for University of California. 1819 is the serial number given to it. So this is a derivative of PBr322 and it is smaller than it, okay, which is approximately 2.7 kb. Okay, now it has your ampicillin resistant gene, okay, and then it has your origin of replication. As you can see, it has lag Z and lag I and multiple cloning sites. Okay, so these are the different genes. I'm sorry for this. So these are the different genes which have been present here. Okay, now what is the next thing we are going to learn is about lag Z and lag Z and the DNA insert. So now remember, what is the lag Z do? This lag Z produces a protein called beta-galactosidase. This beta-galactosidase, it produces blue colonies when it is present on an X-cal medium and an IPTG medium. So when it is present in it, it produces blue colonies. Okay. Now, if you are inserting your DNA at the lag Z site, that means the lag Z becomes non-functional and it is beta-galactosidase deficient. Hence, it cannot produce blue colonies and it produces white colonies on the medium. This is how you can screen for the transformation. Okay, and this is the basics. Okay, so therefore, when appropriate E. coli cells are transformed, they are grown on a medium which has ampicillin, X-cal, and IPTG. And the white colonies, which are also known as the recombinant DNA, recombinant colonies. Okay, these are being selected because they have already been transformed. Okay, so you have another vector or another gene or plasmid, I should say, another plasmid carrying a different genes. Okay, so there are many like PBR327, PUC9, PUC12, PUC13, PBR328, PBR325, PGEM3Z. The PGEM3Z is also similar to PUC vector. Okay, it has same size. It has all lag Z also, ampicillin resistance, and then it has ORI. One thing which it has is it has a promoter of T7 phase. Okay, that is an RNA polymerase. So we will learn more about it. Okay, this was all about plasmid vectors. In the next video, we will be learning about bacteriophage vectors, and don't forget to take your notes.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.